Well, well, first of all, let me just mention, I don't know how many of you know, but someone who was in the department for many years and who was really a wonderful person recently passed away last night. Paxton Tang, who had been sick for a while, died last night. So there will be an email that Marilyn will be sending around. And his wife works in the department as well. He had pulmonary cancer, which had spread. We knew he was ill. He did pass away. From there, it's my pleasure really to welcome David Gamm to the department. As I heard David mentioning to you, he is a pediatric ophthalmologist, which is one of the few pediatric ophthalmologists in the country who's really a clinician scientist and is doing basic science research.
Eric Pierce is called.
And David is facetiously telling him about our big snowstorm that's coming. He spent most of his life in Michigan and Wisconsin.
Yeah.
And, I was in Ann Arbor 2 weeks ago, and about 2 inches was coming down. I was impressed. My God, there's all this snow here, and the news reports, oh, there are a few flurries coming around. Whereas here we would have closed down the city, and it would have stalled everything. So I'm sure he won't be impeded by our weather, although perhaps the airplanes may be a little bit. But David did go to both the college at the University of Michigan as well as medical school there. Left Ann Arbor and went to Madison, Wisconsin, and hasn't moved since then.
No,
he's entrenched in medicine. For those of you who do follow the weather, Madison gets a lot more snow even than Ann Arbor. So it's a very wintry place. David, in terms of his clinical training, went through the residency program in the Department of Ophthalmology at Wisconsin. We have many acquaintances and friends there. As a matter of fact, your current chairman
is that again? Well, yeah.
Yeah, is actually a high school classmate.
Oh, really?
Yeah.
So you hail from? He's from New York, right? Brooklyn. Bronx or Brooklyn?
Bronx.
Bronx.
Bronx. And actually,
he's very proud of that, by the way.
Oh, listen,
I think he makes it thinks he's a little grittier because of that. He's not gritty at all.
The reason he says that is because Bronx High School of Science, which was a co-ed school, which was a very competitive school to get into, was right next to Clinton High School, which was all male, which were really very tough, and they were always #1 in everything sports-wise. And so they always used to intimidate us going to school every day. That's why he's brilliant.
He was the muscle for Bronx School of Science.
That's right. He was the muscle.
I don't know I don't know what that says,
so
but
you can see, you get the picture
yeah.
right away. In addition to that, David is very much been interested in stem cells. He is an investigator in the YSL, the University of Wisconsin Stem Cell Regenerative Medicine Program. Also, an investigator in the Weizmann Center for Stem Cell Research at the university. And he has done some very insightful and very interesting studies looking at retinal regeneration using induced pluripotent stem cells as well as other stem cells. And so we're delighted to have David with us today. He's going to talk to us indeed about induced pluripotent stem cells and their potential for the treatment of retinal diseases. So David, thank you very much.
Thank you very much. And again, I really appreciate the invitation. I've had a great time here thus far, and we'll continue to have a good time talking to folks the rest of the afternoon. It's nice that it's a kind of an intimate environment, so anybody has any questions along the way, please just raise your hand and it won't bother me one bit. So move right on here. So in 1998, at the University of Wisconsin, Jamie Thompson was the first to develop a technique to grow embryonic stem cells in the lab. And so what he did was he took 5-day blastocysts, took the inner cell mass, and devised a way, is that one working? to grow them in culture. And how he did that initially was as these colonies. So these are colonies of thousands of individual embryonic stem cells grown on a layer of mouse embryonic fibroblast feeders. Nowadays, we don't need the mouse embryonic feeder layers. We can use Matrigel. We can use other media constituents that eliminate the need for those feeder layers. But essentially, they're grown the same way as these in these colonies on culture plates. And the key thing about any pluripotent stem cell is it has the ability to, A, self-renew, so it can make more of itself, theoretically forever, although over time and passaging you tend to develop karyotypical abnormalities, so it's not as though we do take one line and carry it on forever. But also it has, to be pluripotent, you have to be able to make derivatives of all three germ lines, so endoderm, mesoderm, and ectoderm. So again, theoretically, it has the potential to make every cell type in the body. Now, practically speaking, there are some cells that are easier to make than others, and some that are very, very hard. People are looking, for instance, for diabetes, for pancreatic beta islet cells. It's a difficult thing to make. Why is that? It's because the cells themselves tend to be biased, and they can be biased either by their very nature or by how you grow them. And so a lot of folks at the University of Wisconsin are looking at different ways of growing pluripotent stem cells, how you maintain them as undifferentiated pluripotent stem cells to try to bias them towards different lineages. As it turns out, neuroectoderm is a default lineage for a lot of these pluripotent stem cells, and that's why we kind of have a leg up on a lot of the other organ systems using stem cell biology. So that's kind of where we were at up until about in 2007, when Yamanaka's lab in Japan and Jamie Thompson's lab again in the University of Wisconsin devised a way to take somatic cells, so fibroblasts, but now can do a lot of other different types of cells as long as they have some proliferative capacity, and reprogram them genetically back to an embryonic stem cell-like state. So because they're induced to become pluripotent, they're called induced pluripotent stem cells, or iPS cells. And just like ES cells, they're grown in exactly the same way. They can self-renew, and they can produce all three germ layers. So they fulfill the criteria for a human pluripotent stem cell. Are they exactly the same? No, they're not exactly the same. They tend to retain some memory of where they came from. So if you derive iPS cells, for instance, from RPE, they have a little bit of an RPE epigenetic background to them. But on a fairly micro scale, they're very similar. So do they supplant the need for ES cells? No, they don't. ES cells are still the gold standard, and we're still looking to make iPS cell reprogramming and technology as close to ES cells as we possibly can. So our laboratory, for instance, uses 1 or 2 ES lines, one of the Bush lines, but we have 30 to 40 iPS lines at any one given time. So in our hands, we do use both. In our lab, we use both. So potential applications. So when iPS cells came along, it added an important tool to our kit. It allowed us to do things that ES cells couldn't readily allow us to do. Of course, it got around some of the ethical issues, which was important, but other things that it brought to the table was the possibility of autologous cell transplantation. So now we have the potential to take any person walking the face of the planet and derive an iPS line from that exact person. So you're kind of reverse engineering it back to a pluripotent state, and then forward engineering it back to a target cell type, which then could potentially be put back into that individual, so so-called customized cell therapy, which may get around problems with immune rejection. I say may because you're doing an awful lot of manipulation when you take that cell, you do the reprogramming, you grow it up in culture, and then if you have to do gene repair like I'll talk about, it's a lot of manipulation. So how similar is that cell ultimately to what you took off that patient in that skin biopsy? So there's still the jury's still out on how autologous it really is, but it's looking pretty promising from the work that laboratories at UW are doing in the brain. So that's one aspect. And the other aspect is disease modeling. Those same people that we can search out and make iPS lines can harbor any disease. So we can look for patients that have specific diseases, no matter how rare it is. If it's on the other side of the world, we have a couple of patients from Turkey that we obtained blood samples from and had them shipped over to Wisconsin. So we can look for people that may be a one-of-a-kind published family in the world, and we can make an iPS line from that family. And therefore, and using that iPS model system, we can either learn more about the pathophysiology of the disease, because as you know, we know an awful lot about the genetics of eye diseases, but oftentimes we know very little, if anything, about the biology of eye diseases. So this gives us at least one way to start looking at the biology or the pathophysiology of these diseases. And then ultimately, we might be able to use the same lines to do personalized drug testing. So if we wanted to for instance, if there's a known drug for a particular disease and we wanted to test dosing or whether that particular person might be responsive to a medication, we could use that person's personalized cells, or we could do drug screening to look for new therapeutics. So from a kind of a cartoon standpoint, what I would envision is to take a patient who has, for instance, a retinal degenerative disease, obtain a skin biopsy, or these days we're pretty much all doing blood samples. You do the reprogramming with a surprisingly few number of genes. So around 4 is all that's necessary, and you can get away with a little bit of, with less than that, actually. And what you're doing is you're inducing those somatic cells initially to express these proteins, but then there's an endogenous switch such that some of these genes that are artificially turned on through the genetic reprogramming process are actually endogenously woken up in the cells. So they start expressing the necessary genes, OCT4, NANOG, on their own. So you do the reprogramming, and at that point you have an IPS line, actually a lot of different lines, a number of different clones that need to be screened, but they still harbor the gene defect. So if at that point your desire is to ultimately get this back into the patient, it probably behooves you to correct the gene defect. So as part of our initial project in our collaborations, we wanted to look at various techniques to try to do gene repair. So we wanted to be able to correct the gene defect in the IPS line and then target their differentiation to the correct cell types that are affected in the disease. And then ultimately the idea would be then to transplant that back into the original patient. However, and that gets a lot of play in the press. That's what everyone talks about, is the transplantation end of things. But there's a great deal of good that you can do potentially with IPS and ESL technology in a tissue culture dish, even if it never goes back into a human. And how you do that is, again, with the disease modeling aspect of things, where you take that same IPS line that harbors the gene defect, targets its differentiation to the cell type affected in the disease, and then do study and testing. So try and determine what is the pathophysiology of that disease. Test various drugs. You can do high throughput screening if you can do it efficiently enough, and then perhaps develop therapeutics that can go back into the original patient. So there's 2 arms then. You can either do a transplantation arm, or you can do something totally in vitro, looking for new, more conventional therapeutics. But there's a huge number of limitations. There are always more limitations than there are advantages to any particular system, and it's important to know what those limitations are so you apply your techniques correctly and you don't end up doing an awful lot of work and not being able to answer the question that you sought out in the beginning to address. So limitations of IPS cells: 1, time and expense. It's expensive. You have to screen a lot of clones. Generally you have to have a number of controls when you're doing these experiments. It takes a lot of person power to do it. So at this point, and the reagents themselves are expensive as well. The process of reprogramming has been shown to introduce, on occasion, deleterious mutations. So by creating the IPS line, you generally introduce anywhere between a half dozen to a dozen or more mutations in the coding region of the genome, of which most of them are silent, not thought to be deleterious, but a couple of them can be. And so that's always something that's kind of in the back of your mind. That being said, any cell culture system probably has those point mutations in coding regions. We just don't look with a high enough powered microscope with whole exome sequencing to find out. With IPS cells, everyone was really nervous about it, so the bar was raised really, really high. So I think it's something we need to be concerned about, especially with regard to transplantation, but it's not a deal breaker by any means, especially when you're looking at specific differentiated cell types where a lot of these genes are not even expressed. That are more commonly found to be mutated. And it's also not necessarily the case that they're mutated during the process of reprogramming. They also could be selected for from the original somatic cell population that you used to create the IPS lines. There is a good deal of variability in lineage-specific differentiation. So for the most part, when we differentiate from ES or IPS cells, the cell types that we get at the end are very, very similar. We can't tell much of a difference, if any difference at all. But the efficiency with which you derive retinal cell types can vary greatly from one line to the next, even from the same patient. And so that means you have to do a lot of screening, and we've developed ways to do that to try to shorten that process down. Then ultimately, if you're going to do modeling or you're going to do transplantation, you need to be able to isolate your desired cell types. It doesn't help you all that much if you've got 2% of retinal cells in a sea of bone and cartilage and everything else. What are you going to do with that? You can't, there's too many confounding variables to do in vitro testing, and there's certainly too much crud going on in there to put it into an animal model system and hope to get something out of it that you can interpret. So being able to isolate the desired cell types is a big deal. And there are there are some ES lines that are available, such as H1, that are pretty darn efficient at making retina if you apply certain protocols. So you can get 80 to 90% of the whole population to be close to all being retina based upon the literature. In my hands, I can't get it as good as Dr. Ray can, but there are lines that you can use off the shelf that are just good lines. With IPS lines, when you're making them de novo from an individual, you don't have the luxury. You have to do all that yourself. And then lastly, you have to be able to recapitulate some portion of the disease phenotype. Are you hoping to recapitulate the entire disease in a dish with a single cell or a couple of different cells? No, you can't do that. But can you boil it down to a manageable piece of the disease that you can look at under a reasonable sized microscope and get some answer that you're looking to get? So you need to be able to not only get that cell type, but that cell type has to have a level of maturity in the system that you're looking at it in, such that it can behave the way it should behave in vivo, and you can get useful data from it. And so I'm going to concentrate on, for the first part, the next part of my talk, on the work that we've done in my laboratory, developing the technique and the method to be able to produce and enrich for retinal cell types from ES and IPS cells. But most of what I'll be showing you is from IPS cells, as well as to do some functional characterization of those cell types.
Before you go there,
Yeah.
Not a very intelligent question, but this reprogramming thing, does it happen naturally by bugs or this and viruses?
Nope, not that we know of. Not that we know of. So there's no, natural viral infection that induces pluripotency. So.
And what kind of evidence we have that once they differentiate and you put in some tissue that their differentiation is done? They don't go back and forth.
It's a good question. So you can follow these. For instance, if you're doing xenografts, you can have human nuclear antigen that you can use, that you can follow them post transplantation. And we're also realize that you have to crawl before you walk. So the first thing you're worried about is that there are some rogue cells that are not fully differentiated that are going to cause a tumor or cause problems, right? Once we've seen postmitotic cells, so that express postmitotic markers, we have not seen them revert back to a proliferative state. So in other words, once we have no more proliferation in a culture or in an animal model. Now, animal models are a whole different situation too, because you're also dealing with immune rejection. So you've got this balance between cells dying and cells living, or in a little bit of proliferation. So I won't go so far as to say we know anything in terms of what could possibly happen, but there's been no evidence to suggest thus far that a terminally differentiated ES or IPS cell can revert back without further genetic manipulation. So the method that we've developed over the course of the last 3-4 years is actually very simple. It may seem a little complicated from what I have down here in the box. For the most part, we don't have to do this small molecule inhibitor step unless it's a really recalcitrant line. But about 2/3 of what makes the protocol work in our hands is actually the mechanical manipulation and the enrichment that occurs through the mechanical manipulation of the cells, as opposed to the media that we use. The media is important, but you can actually use a very minimal media and still get good neuroectodermal, forebrain, and retinal efficiencies because of the default nature of these cultures towards a neuroectodermal fate. So we take the initial ES cell or IPS cell colonies, raise them into embryoid bodies. That gets them away from any of the feeder layers that they have and initiates the differentiation. We also eliminate FGF. Plate these back down after a period of time in the presence of what we call a neural induction media, which is predominantly N2. And this is kind of where the magic happens here. Over the course of a period of a week to a week and a half, these will start to grow into one another, and we'll start to see what we call neural rosettes. It's almost like primitive neural tubes. Now, the mechanical property that we take advantage of is the fact that these clusters here are loosely adherent to the underlying plastic. These other surrounding areas are very firmly attached. So we can just kind of blow media across these, and these will pop right off, like the yolk off of an egg. And so there'll be a little hole right here. And actually, these tend to make RPE. But once we pop these up, we wait another 2-3 days, and we see a couple of different populations of what we call neurospheres. There's a more uniform neurosphere that's a little darker, and then there are these kind of golden Cheerios, these vesicular type structures that have a neuroepithelial rim to them, and they're hollow. And so I'll skip a lot of years of work. We did a lot of reporter lines when we were trying to look for the retinal structures, and it was actually staring us in the face the whole time. So eventually, we said, well, let's just pull these suckers out because they're easy to do manually. You can just tell them under light microscopy and then pool them together. When we did so, and then sectioned them, we found that they were 90+% expressing the progenitor marker for retinal progenitor cells of CHEX10 as well as PAX6, Ki67, SOX2. If we follow them over time, so here's an initial one of these little guys. We're growing these in batches, so we can have hundreds of these in a single flask. So here's one of them here. If you follow them over time, so day 20, and by the way, the timing and sequence of this is all very similar to what you would expect in normal human development. You have to add 5 days on because you're taking it from a 5-day blastocyst initially, or that's kind of the stage that you get it at. But it's very similar to what the timing that you would expect in normal retinal development. After a short period of time, we begin to see a bit of a bilayered structure where we start to get expression of BRN, other ganglion-like markers on the inner portion of it, along with this CHEX10 positive kind of neuroblastic layer on the outer portion of these structures. And as you can see, the ganglion-like cells don't have any place to go, so they elaborate long processes that are beta-3 tubulin positive, but they don't really have much of a place to go. And eventually, they tend to sneak their way back out and wrap around the outside of these structures, which is not normal and tends to contribute to the disruption of the laminar structure. And so what happens in those cases, and here's some expression of calretinin, so we see amacrine-like cells, QCD positive, BRN3 negative cells too that are lining the inner portion of these structures. But the majority of them, probably 75% or so, eventually lose this primitive bilaminar structure and start to form rosettes, where you have photoreceptor-like rosettes surrounded by ganglion-like markers. Here it's TUJ1 in blue and BRN in red. But on occasion, and we're still working on why it works and why it doesn't on some occasions, you'll maintain this laminar structure and actually will become more structured over time, such that by day 72 and upwards of day 100 in culture, we start to see expression of recoverin, other photoreceptor-like markers, and we see this three-layered structure where we have an outer nuclear-like layer that harbors the photoreceptor-like cells. This intermediate layer will have CHEX10 positive cells. We'll also have PKC positive, PKC alpha positive, bipolar-like cells, and then we maintain this inner layer here of BRN3 positive, TUJ1 positive, QCD1 positive cells. So to this point, that's about as advanced as we can get this laminar structure to occur. And we're getting better at reproducing this from line to line and getting a higher percentage to do it.
Excuse me, what was the original stimulus for this type of differentiation other than other brain neurons?
There's no brain neurons at all in these. These are isolated optic vesicle structures that have no other forebrain in them and no RPE. So this is default. So these are being kept over time in B27 supplement, and so this is just, it was what they'll do on their own. So in other cultures, in IPS and ES cells, it's the same way with Tom Ray's work and with Takahashi's work. If you separate them out, it's a default program for a lot of them. What's novel is exactly what they can do in the absence. Like in Sasai's case, there was forebrain there with the optic vesicle structures, and I'll get to it in a minute. And the benefit of that is you can induce RPE formation and ultimately form optic cups. We don't see optic cups and we don't see RPE unless we manipulate them, and I'll show you that.
You don't see Mueller cell?
Yes, we do. I'll show you a little bit later on. They're quite, Mueller cells, most of what we see are biased towards early-born cell types. We see cones, we see ganglion-like cells. Mueller glia, later-born types, bipolars, are a minority population. They come about, but when they're kept in suspension like this, Mueller glia remain a minority population. However, if you plate them down, the Mueller glia are proliferative, and they will quickly take over the culture. One of the reasons we like to keep them in these suspended structures is we prevent the Mueller glia from taking over. But we also benefit from the Mueller glia being there for synapse marker expression, which I'll show you in a moment. So again, this is kind of over time, gene expression of various markers of different retinal cell types from day 20 to day 110 of differentiation. Here are some broken up optic vesicle-like structures plated down. We're also looking here at relative fold change in expression of these markers over time. We can see CRX positive photoreceptor precursor-like cells are the predominant population that we see in our cultures. So, as I mentioned a little bit ago too, we also see synaptic marker expression relatively late in culture, and it coincides often, well, at least the protein expression coincides with the appearance of glial cells, so S100. We don't see CRAWL-BP expression very well in our cultures, but they'll be GLAS positive. They'll also express GFAP as well. And when we start to really see some of the synaptic markers come on, it coincides with the expression of some of the thrombospondins, which are released by glial cells and promote synapse formation. So we see both presynaptic and postsynaptic receptors. We see pre and postsynaptics that are synaptic markers that are co-localized, for instance here on ganglion-like cells, and then on some of the, we also see connexin 36, some gap junction markers, although those are relatively sparse in the cultures.
You said you dissociated them as well.
For this particular one, in order to see these, we tried doing it in slice cultures, but it's too messy. So to get the synaptic marker images that we got here, broke up the spheres themselves, plated them down overnight, and then did the staining thereafter.
So when you break up the spheres, do you see a change in the term of differentiation? That like the cell population?
Yes, you do. You do. So if you
must probably are affecting each other differently.
Right. So if you break them apart, well, it depends on what you do. If you break them out into little chunks, then you can go a couple of weeks and it doesn't really affect the differentiation timeline very much at all. If you dissociate them fully, which is hard to do when they're very old, by the way, they lay down a lot of ECM, and so when you pull them apart, you have to be very gentle, otherwise you just rip the cells apart. But if you do it early enough, what you'll do is you'll have a period where it's delayed. So it'll be 2, 3, 4 weeks where you'll see very little differentiation, and then it'll start to pick up again. And so it just takes a little bit longer to get to these various points. But we've done a lot of manipulation because there's benefits to having it in a monolayer culture, and there's benefits to having it in these arrangements where they have various cell-cell interactions. So from a functional standpoint, we've looked a little bit as well at doing whole cell patch clamp onto the photoreceptor-like cells. So the recoverin positive, opsin positive cells, which are very readily identifiable in culture. So they tend to have a little nub and a little dunce cap on one side, which expresses opsin, and then it has a longer process on the opposite side. So they're polarized. And so we can tell if they're on the edge of these cultures, we can kind of tell by their morphology whether or not they're going to be a CRX positive, recoverin positive, opsin positive cell. So we've done whole cell patch clamp on these cells and single cell PCR, and they have a lot of the machinery that's required for phototransduction. They don't respond to light. They don't have outer segments. No cell culture system that's dissociated cell culture system can support the formation of outer segments. You can get them in explant cultures, but not when you dissociate them. In effect, that's what we have, is a dissociated cell culture system. So it's not surprising that it doesn't respond directly to light. However, in its resting state, it's kind of a light-adapted cell. So it has an outward positive current. It's with delayed rectifier potassium channels. And interestingly enough, if you incubate it or expose it to membrane permeable bromine cyclic GMP, 8-bromine cyclic GMP, which then gets into the cell and it essentially mimics the dark state, you'll see a very rapid change in current. So you start having an inward current, and you'll get a switch from a membrane potential that is akin from the light-adapted to the dark-adapted state, which suggests that at least some of the phototransduction machinery is present within these cell types, although essentially there's going to be a limit to it. But it does allow us to say, for instance, set the bar as to what we can model. Can we model something complex like an outer segment-based, rhodopsin-based RP? No, we can't. Could we perhaps model something like the PDE6B mutations? It's possible, as long as you had a very discrete question that you were asking. Getting back to the RPE question, so we don't see RPE in these cultures, and that's not surprising. Tom Ray, when he did his work in chicks, would isolate optic vesicles from chick embryos. Now these are vesicles that would normally invaginate and form a bilayered optic cup with an outer layer of RPE and an inner layer of neural retina. However, if you just separate those optic vesicles out and grow them independently in culture, you don't get RPE. You need signaling cues from the head mesenchyme to promote RPE development. You can, so in his initial study, what he did is he replaced the need for head mesenchyme. So if he co-cultured his chick OVs with head mesenchyme, he would get RPE. However, he could replace the head mesenchyme and use activin A or BMP or other factors that were known to be secreted by head mesenchyme. And that, in fact, works also in the ES and iPS cell cultures as well. If they're exposed to diffusible growth factors that are normally released by head mesenchyme, we can produce a layer of RPE on the outside of these structures, separate them out, plate them down, and get a monolayer of RPE. And they also demonstrate the functionality that we would expect from a cultured RPE cell, similar to what the gold standard is for fetal human RPE. They establish correct cell polarity with expression of ezrin at the apical surface and bestrophin in the basolateral surface, or near the basolateral surface. We do FURA2 imaging and look at calcium responses with ATP as a trigger. We'll see appropriate responses to ATP. We get tight junction formation with ZO1, occludens, phagocytosis using FITC-labeled photoreceptor outer segments, and then also doing electrophysiology with perforated patch clamp. We can see ion conductances that are similar, if not identical, to what we get when we do the human prenatal RPE. So that kind of sets up then the system. So what at this point do we think we can model? Well, RPE is obviously the lowest hanging fruit in all this. RPE has been used for years and years and years as a model system in culture. It performs a lot of its functions in the absence of any other cell types. So it's an ideal cell type, not just in the eye, but in the body, to do iPS modeling. Because you don't need complex interactions from other cell types or other tissues to ask some pretty in-depth questions with regard to cellular function. So we wanted to direct our attention first towards RPE-based diseases, and I'm going to show you two that we've done, one that we've published and one that we have not. So the first disease that we looked at was gyrate atrophy. So this is a rare autosomal recessive disease, primarily involving the RPE, although it does have some minor effects elsewhere in the body. Fairly ubiquitous enzyme that's affected in this disease. It leads to blindness by the 4th to 5th decade of life, and it's caused by a lot of different mutations in the ornithine delta aminotransferase gene. Now this particular gene, the OAT gene, is again expressed ubiquitously, but it's expressed at a particularly high, probably the highest level in the body in the RPE. This gene it's a matrix mitochondrial protein. It's involved in the production of proline and glutamate, so it's a pretty critical protein. It also has a cofactor, vitamin B6, so it requires vitamin B6 in order for it to function correctly. And there's a small subset of patients with gyrate atrophy who will respond to vitamin B6 supplementation by delaying the progression of the disease. So we obtained a skin sample, or a fibroblast sample, from a 27-year-old woman with a particular mutation in the OAT gene. This woman had already been tested for vitamin B6 responsivity, and it was deemed unresponsive. But recall how you actually test this. You can't reach in and test the patient's eye. You can't test the patient's RPE. So you give them a challenge for vitamin B6, and you measure plasma ornithine levels in their blood. Or you can take a skin sample and you can test directly their fibroblasts. But you can't test the RPE. And the RPE is really the only place where this disease is manifest. So there's got to be something particular about the RPE in this disease. But there's no way to test it directly. Derive the lines using lentivirus, as well as transgene-free episomal techniques that we also use under defined culture conditions. And then used our protocol to derive RPE. And the nice thing about RPE is not just that it recapitulates the physiology so well in culture, but I can expand it. So you can get a portion of this, you can grow more and more, I can expand it, I can get a lot of material to work with, as opposed to something like, say, a photoreceptor cell, which is post-mitotic.
How do you maintain the differentiated status when you're expanding it? Because as you know, a normal RPE cell, it's upon expansion, we undergo EMT and differentiate.
So you could pass them 2 or 3 times, but that's it.
So it's only
yeah, it's not forever. It's not a transform line, but being able to expand them 1,000 to 10,000 fold, boy, if you could do that with photoreceptor precursor cells, it'd be a lot easier to work with. But it's a good point. When people say they're passaging them 8, 9, 10 times, check the karyotype. There's something up.
I didn't get it, but I'm sure these cells express RPE65.
Yep, and I'll show you. Yep.
And they phagocytose out to segments.
Yep. Yes, they do. That was on the previous slide that I showed you, but I'll also get into that with the next disease model that I'll show you. They do it quite well. So in this particular, one of the reasons I picked gyrate atrophy is not because we were going to save the world from the scourge of blindness, because it's very, very rare, but because it had a very discrete assay. So the OAT enzyme has a colorimetric assay, so I can not only follow the genetic defect in these lines to make sure that the patient cells has the defect and the iPS cell has the defect and the RPE that's differentiated has the defect, but I can follow functionally the OAT enzyme defect as well. So, and what we found was that in normal wild-type human fetal RPE, as well as in wild-type ES RPE or a wild-type iPS line that we see relatively normal levels of OAT activity using this colorimetric assay, and we had negligible, if any, OAT activity in our disease line. Now, that was with, however, normal levels of vitamin B6, which is always included in these assays. Now, when you do the challenge test for vitamin B6, especially if you're doing it with skin cells, you then add a great deal more. And we did a dose response, but I'm going to show you the 600 micromolar amount of vitamin B6, which is traditionally used for the fibroblast testing. And we found that in this particular patient, we in fact restored normal activity for the OAT enzyme. And this patient's gene defect is actually in the vitamin B6 cofactor binding site. So it kind of makes sense, and it had been published in animal model systems to be responsive to vitamin B6, but this woman was told that she was not, and was taken off of the vitamin B6. So here's an example where, with extremely exorbitantly expensive personalized IPS testing, you can actually look at that person's RPE, and it gave a different result than what the serum testing and fibroblast testing gave her. Last thing we did with this particular, on this particular study is to look at the idea of gene correction. That's another reason why I picked gyrate atrophy, was I wanted such a strong readout that when we did gene correction or gene repair, that it would be unambiguous that we'd get a functional improvement or a functional change. So in collaboration with Jamie Thompson's lab and Sarah Howden, who worked both in my lab and in his lab, we derived this BAC-mediated or bacterial artificial chromosome-mediated homologous recombination approach to essentially swap out one of the good genes, because it's an autosomal recessive, so you really only have to target one of the alleles. As it turns out, about a third of the clones that we got that were gene-targeted actually had both alleles targeted and were corrected. We found that by sequencing that the mutation had been corrected by FISH, by array genomic hybridization, by whole exome sequencing, by karyotype. Everything else was normal. And this was relatively a remarkable result in my mind, because there was a number of different steps that occurred during the gene repair process, and there were no new mutations introduced throughout that entire process. The line still had a handful of mutations from when it was originally reprogrammed, but the gene repair process did not introduce any other gene defects into the line. And as we would expect, after gene correction, the OAT activity was corrected.
What was the frequency of that?
It actually was not terrible. It was about 1%. So this is, and you have a drug selection. So it's not hard to pick these out. The bigger issue was the targeted selection. So only about 15 to 20% of the recombination events were actually targeted recombination events. So a lot of them you'd see not just 2 little spots of FISH, you'd see 3 or 4, right? So it's not actually targeting the gene, the locus for the gene, but actually randomly integrating somewhere. So that's what you have to be careful of. And the array CGH was very important for us to quickly screen and make sure that we didn't have too many copies, for instance, of the OAT gene. All right, so,
Before we go
yeah.
on, so did I hear you correctly? You showed that with 600 micromolar B6, that in fact you were able to have her, quote, defective RP respond. But that her fibroblasts would not with 600 micromolar?
No. So we tested the same patients because we had their fibroblasts
So
too,
you
right?
tested the
It did improve. It did improve. It was a slight increase, but not. It was about 10 to 15-fold less than what we saw with the actual RPE, which, if you look at the level of expression of the enzyme, might very well. So you basically have a greater sensitivity with the.
Do you have any insight into why there was selective sensitivity?
You have a much higher expression of OAT in RPE. And also, there's got to be other factors too if you think about it. Why this is a critical enzyme, right? It's your whole body, but you have no other manifestations of it. So there's got to be probably some redundancy in other cell types. There are no skin defects in gyrate atrophy. So there's probably some compensatory mechanisms for it that aren't present in the RPE. But that's a good question, and what I'm not mentioning is what we're still doing with the gyrate atrophy line. So we really haven't, the whole idea of the pathophysiology of why the RPE is more sensitive than the rest of the cell types is another good way to do this. And we have fibroblasts and RPE to juxtapose against one another.
And is there any evidence that there is regional difference in sensitivity within the RPE? In other words, we all know gyrate atrophy starts, as most retinal degenerations, in the peripheral retina, and then sequentially progresses posteriorly. Is there any difference in
Well, we know that there's, as you well know, that there's a lot of differences in the RPE itself regionally. So one could easily envision that there's different susceptibilities, but nothing that we know of. Whether or not there's higher amounts in the mid-peripheral RPE or, yeah, that we don't know of. Because unfortunately in those situations, or fortunately, we have like David Ballet's models where, but they're mouse, so they don't have a mid-periphery and a macula. So now this is something that perhaps with adaptive optics and some of the other heavy ion work that's being done, that maybe people can get a better handle on, but not that I know of. Okay, so as I mentioned then, what that last study then did was just proof of concept. It was one of the two first IPS modeling papers that came out in retina, the other one being for RP that looked at cell death in rods. But what we wanted to do is kind of proof of concept that you could do gene repair, you could do targeted differentiation from them, and that you could do, you could study them in a dish as well. So you had the ability to make a positive impact, both making a step towards transplantation, but not obviously there's a huge black box still here, and also working with them in vitro. For the next disease that we wanted to look at, and the only other one I'm going to talk about today, we wanted to raise the bar a little bit higher. I wanted to pick something that was still RPE based, but had a little more lack of knowledge in terms of the pathophysiology of the disease. So you can only go so far saying, okay, it's gyrate atrophy, it's got an OAT enzyme defect, and the IPS cells have an OAT enzyme defect. You really want to be able to, at some point, shed some additional knowledge onto a disease or disease process or the ability to treat that particular disease. Now, IPS cells, as I've talked to people already, don't stand on their own. They're a tool. Just like animal models are a tool, observations of patients are a tool. All these things have to come together to hopefully improve our knowledge of the biology of a disease and lead us towards a treatment. So in talking about IPS disease modeling, it's not a standalone process. It's something that you have to meld with the available data that you have. And it's very important for me in modeling diseases with IPS cells to start with what we know in animal models and say, well, what's the shortfalls in animal models, and how can IPS modeling perhaps bolster up some areas that are a little bit, that the field is a little bit unsure of. So for this, we chose Best disease. So Best vitelliform macular dystrophy, second most common inherited macular dystrophy, second to Stargardt. Slowly progressive with onset typically in childhood, but sometimes later. Very variable course and phenotype. There's an intrafamilial difference that you'll come across that's pretty striking sometimes. The diagnosis is based on fundus, a very typical fundus appearance, EOG with a difference in the Arden ratio, family history since it's autosomal dominant, and these days more and more just straight genetic testing. And it's characterized by the appearance of this subretinal vitelliform lesion. It's kind of this yolk-like lesion. And they don't always progress the same way, but you have different stages that are recognized, including a pseudohypopian stage, a scrambled egg stage, and then more of a gliotic scar stage as you start to get secondary photoreceptor degeneration. And you get accumulation of this yellowish substance, or lipofuscin, which is an abnormal shunted pathway of photoreceptor outer segments that produces this substance that can neither be removed or degraded. And so it's toxic, and it can also be further manipulated to become something called A2E, which is a very toxic molecule. And it accumulates both within the RPE as well as in the subretinal space. So what do we know about Best disease? Again, autosomal dominant, caused by a host of different mutations in the BEST1 gene. It's expressed in a more limited, has limited expression, predominantly in the RPE. It's also in the respiratory epithelium, so it's also in some other epithelial cells. And essentially, the thought is that you have a perturbation in BEST1 function of some type, which through a mechanism that's not completely known, ultimately leads to altered handling of photoreceptor outer segments. Okay, so the RPE's one of their main job is to ingest 10% of the outer segments of the photoreceptors on a daily basis. And these outer segments are filled with photooxidized proteins and lipids. And so the RPE has to handle this tremendously violent and toxic environment and compartmentalize it, diffuse it, and get it out of the way. So it's a tremendous job that it has to do that pretty much no other phagocytic cell has to face on a daily basis. So once you have this altered outer segment handling, if it doesn't occur correctly, you could have essentially a backup in the system, leading to this accumulation of this indigestible material, this toxic material, and ultimately causing photoreceptor death. Interestingly enough, however, the RPE survive. So in postmortem studies, the RPE is not degenerated. So from a cartoon standpoint, what they're faced with is gobbling up these outer segments into what are called phagosomes. These then fuse with lysosomes, which lower the pH, make it more acidic, also release enzymes, and form this what's called a phagolysosome. And if it's done correctly and it's broken down correctly, then you can eliminate the products of the photoreceptor outer segment. With lipofuscin accumulation, what happens is something goes awry here. Now, where it is, it can be probably in more than one position, but something goes awry during this process, leading to the formation of these products that are no longer that the RPE is no longer able to deal with, and they build up, and eventually they'll build up in the subretinal space as well, forming those vitelliform-like lesions. So what can go wrong to lead to this? Well, it turns out that this process is extremely complicated. It can never be easy, and it's controlled by a number of different processes and ion conductances within the RPE cell, of which chloride is one, calcium is a big player, and then bicarbonate and pH regulation. So all of these can play a role in this process. So what don't we know about Best disease? Well, we don't know much, for sure about its location, subcellular localization, and we don't really know what its function is. It's been postulated to do a number of different things, including being a calcium-activated chloride channel in the basolateral membrane protein, so actually in the basolateral plasma membrane, at the bottom of the RPE, a sensor for volume regulation, a bicarbonate transporter, and also a regulator of sodium-hydrogen transporters and channels, a cytosolic, so those are all in the plasma membrane, cytosolic regulator of basolateral membrane, so not actually in the basolateral membrane itself, but associated with proteins in the basolateral membrane, and where it regulates calcium channels or calcium-activated chloride channels. It's also been suggested and shown in some systems to be actually in the endoplasmic reticulum, where it can function perhaps as a chloride channel or a calcium sensor there. So we took 2 patients with Best disease and 2 unaffected siblings, a 52-year-old male and a 62-year-old male, with different mutations. The first one here is in this putative calcium binding arm, and then the second mutation is here in this cytoplasmic loop. And I took different ones on purpose because the phenotype is very similar in these patients, although the path that they get to that phenotype can vary. And so the idea then is that the pathogenesis has to be at some point be relatively similar between all these patients. Both of them had poor visual acuity, and as I said before, we had the unaffected siblings. Derived the IPS lines, as I mentioned before, made sure that the point mutations were maintained in each one of the affected lines. And then, as I mentioned with the gyrate atrophy, we could direct them using our system to RPE. The RPE could be expanded, placed on transwells. And you can see the difference between the prenatal RPE, one of our control lines, and the BVMD. And so we have 24-well plates that we produce in these transwells or on cover slips that we can use to do all of our studies with. Characterized these lines, and at some point we want to see that they're very similar. So if there were things that we wouldn't expect to be different between the lines, that would make us raise an eyebrow. But their expression of typical housekeeping genes for RPE were the same across all of our lines. The expression of Best protein was the same in the control and affected lines, as well as RPE65. And that's been shown before that the Best mutation does not lead to a haploinsufficient state. There are similar levels of Best protein made, so it's not as though the Best protein isn't made in the face of the disease. Cell polarity is maintained in both the control and the wild type. And then, as I mentioned before, we get good tight junctions. So here is a measurement of transepithelial resistance. So we actually look to see how well these form tight monolayers and with tight junctions between the cells by measuring the resistance across the RPE monolayer in these transwells. And 600 is a good transepithelial resistance that shows you've got a good tight monolayer. And with passage 1, we're able to achieve that with all of our lines, and we can do actually up to passage 3. But if we get much past passage 3, they tend not to proliferate enough to reform tight junctions. So we tend not to go past passage 2. So the assay that we looked at first was phagocytosis. We wanted to see, was there a difference between the affected Best disease lines and the control siblings, as well as fetal RPE, when they were challenged with fluorescently labeled photoreceptor outer segments. So the paradigm at first was to feed them for 2 hours and then wash that out and measure at 0, 4, and 24 hours. So in our control lines here, and there are error bars on there too, but in our control lines, we set that at 1. And when we looked at 4 hours after feeding, we actually saw slightly more label inside of the RPE cell in the Best disease patients than we did in the control, although it really was not statistically significant. So it didn't appear as though the uptake, at least, of the outer segments was being affected. At 24 hours, which by this point you should have well cleared out all of your outer segments because you've got to get ready for your next feeding if you're a wild type, we found that there was a significant difference in the amount of FITC labeled photoreceptor outer segments in the Best disease versus the control. That's one way to look at it. Another way we wanted to look at was rhodopsin degradation. This is a very classic way to look at POS handling and degradation of proteins within POSs. So feeding the cells a relatively light or normal meal of 20 photoreceptor outer segments per cell. With the control lines, we see that within 24 hours, or between 4 and 24 hours, we see elimination of all rhodopsin, the monomer, and the dimer. Whereas in the mutant, we see it hang on even up to a little bit by 48 hours. And again, you want to be able to clear this for the next round of feeding. If we challenge them a little bit more and give them a slightly bigger meal, we'll see some retained rhodopsin in the controls, but now we see it in the mutants all the way out to 120 hours. So there does appear to be a delay in that. Another experiment that we did was to feed them unlabeled photoreceptor outer segments. So now we're looking just at autofluorescence, presumably lipofuscin or lipofuscin-like compounds. So we fed them the heavier meals for 3.5 months straight, and then washed it out and looked 2 weeks later after. So we washed them continuously for 2 weeks and then looked at them. So the controls would have some accumulated autofluorescence, but comparing that to the disease lines, we see significantly more autofluorescence in the disease lines. So one might think then that having additional buildup of lipofuscin or other toxic byproducts from photoreceptor outer segments would set up a situation where you'd be under some stress. And in fact, that's presumed to be part of the mechanism of the disease, where you get into this vicious cycle where you have accumulation of lipofuscin, A2E, increased oxidative stress, which then leads to even poorer handling of photoreceptor outer segments, more stress, and so on and so forth. And so we looked just recently, this is our first experiment, looking at protein oxidation in our cells, and we found that this one here was not significant. We'll see, we'll do some more experiments with it, but the trend was there. But the difference here between the control and this particular mutant was significant in terms of higher levels of protein oxidation in the Best disease lines. So it gets down then to mechanisms. All right, so we think that we have a phenotype, which was we were happy to see that there appears to be a difference in the handling of the degradation of rhodopsin, the handling of photoreceptor outer segments. But why is there a difference? And this is still a work in progress. So I'll show you what we've done so far and tell you what we're continuing to do. Again, just to remind you that this is a process that involves a great deal of potential involves ion regulation, pH regulation, and disruption of any of these factors could lead to a problem with photoreceptor outer segment handling. So again, where is Best 1 interacting here? And this is just to show you how complex it is. This doesn't even show the ER. It's very difficult to try and say, all right, where are we going to start here in terms of what Best might be doing in this particular system? So what we decided to do is first look at localization. So let's just step back and just try and get an idea of where the Best protein is localized. And so when we do that, we see it predominantly near the basolateral membrane. It looks very similar between our control and our mutants. This might look a little different, but if you pan the entire culture, they're very, very similar. We then did membrane fractionation for both the wild type and the mutant. I'm not showing the mutant here right now because we haven't repeated it, and so I'm not going to put myself out and tell you what we found there yet. But interestingly enough, what we saw was that we did not recover best protein in the plasma membrane fraction at all, or the mitochondrial or lysosomal fraction, but instead in the microsomal or the ER fragment fraction, as well as in the cytosolic fraction, which Chris Hartzell and a number of other labs have also corroborated in pig models and other systems where when they do biotinylation of whole cells, they don't label the best protein. So this supports work by Marmerstein and now Hartzell as well that suggests that best protein is not actually in the plasma membrane itself, but rather associated with it. And we think it's associated with it through this molecule STIM1, which is a calcium sensor. So that would be the cytoplasmic fraction. The microsomal fraction in the ER then suggests it might also play a role in ER ion homeostasis. So we looked also at co-expression with calnexin, which is an ER marker. And certainly it's not completely overlapping, but we do see co-expression, partial co-localization of BEST1 with the ER marker calnexin. And we're doing co-IPs and other experiments as we speak. So based upon just the localization data then, one might be able to say, okay, well, it doesn't look like it's in the plasma membrane, so perhaps these are less likely to be part of the mechanism of BEST1, and thus less likely to be part of the pathophysiology of BEST disease. And then whereas these potential functions look perhaps a little bit more promising. And of course, none of these are mutually exclusive either. So it's not as though you can only do one thing, and the likelihood is that it's not just doing one thing. So the last assay that I'll show you then is to look at calcium. So we then turned around and said, well, okay, we don't think it's a basolateral membrane protein. And a lot of what we're looking at tends to point towards an effect on calcium and calcium handling. So what we did is we took, first in the IPS lines, the control lines and then the disease lines, and challenged them with ATP, which causes release of intracellular calcium stores from the ER. And this has also been done in some overexpression studies by Hartzell and Marmerstein. And so what we're seeing is very similar to what's been shown in a couple of other model systems, but not consistently across all the model systems, where we see a higher peak release of calcium and a slower uptake of calcium in response to ATP. And that's with both of our mutant models. To test this further, we also have lentiviral constructs that overexpress the wild-type BEST gene and the mutant BEST gene. And so we overexpress those in the fetal RPE. So a lot of the experiments, I didn't show you everything, but a lot of the experiments that we did not only in IPS lines that endogenously express levels of BEST, but we also overexpressed these gene constructs in human fetal RPE to look at differences. Because oftentimes what you see is you'll see the same trend, but a much larger difference. In this case, when we overexpressed, and this just shows you that we get similar levels of expression in both our mutant construct and our wild-type construct, and this is the endogenous level of BEST1, we see, again, a larger peak and a slower uptake of calcium compared to the wild-type overexpressed BEST protein.
Can that be modulated by B6?
So by B6, I don't know. And your thought being?
Well, I'm just wondering whether you use that as a challenge assay. No, hang on, you switched diseases, sorry.
Yeah, I switched diseases on you. Yeah, it's a lot of stuff I shoved in here, probably too much. But so what are we doing ongoing? We're doing perforated patch clamp. I can tell you, as of the tests that we've done so far, we do not see a gross difference in chloride conductance, at least from perforated patch clamp. Further examination of oxidative stress and autophagy. We're measuring intracellular and lysosomal pH. And then we're doing the co-IP studies to get a better handle on what the complex is that BEST1 is involved in. Presumably near but not in the basolateral plasma membrane as well as in the ER. And then ultimately what we think we can do is developing these assays is not just helpful to us in terms of trying to understand the pathophysiology of the disease, which I think that the model system does allow us to contribute to. But also the more we can manipulate and determine assays that are useful and relatively easy to reproduce, we can then start using our robotic cell-based screening systems that we have access to at the University of Wisconsin, where we can do completely enclosed ES and IPS cell culture and screening with drug libraries and an assay system as well. So again, RPE is good for this because you can expand it to a limited extent. You can get a lot of different wells in a system. It's relatively easy to maintain in culture, even with a robot. And then we can screen libraries that we will pick based upon what we think the pathophysiology will most likely bring out as the most logical way to introduce therapeutics. So hopefully then what I've talked about today is not so much, or equal parts of what can IPS cell modeling potentially do, but just as much what it can't do, what the limitations are. The fact that it doesn't stand alone, just like no system stands alone. It has the opportunity to advance our knowledge. It gives us a human-based system. It gives us a system that's taken directly from patients, so we can look at that patient's retina. We can say, this is this patient's clinical course, and here's the RPE from this patient. Now, it's not an aged RPE either, unless, right? So you're basically turned back the hands of clock. So you're not taking out that person's diseased 80-year-old RPE, right? You're taking out something that's presumably much younger. How much younger? What do you have to do to manipulate it to get it older? I mean, a lot of people are looking into this not just for retina, but also for modeling Parkinson's, modeling Alzheimer's. So this is a huge area of research in the IPS field in general to try to model these age-related disorders. So we're somewhat benefit of looking at relatively early onset diseases. Since I'm a pediatric ophthalmologist, I don't have to worry about an 80-year-old, but worrying about an 8-year-old can be just as bad if you have to wait a long time in culture to do your assays. So I'd like to acknowledge just the folks in my lab. Joe Phillips did a lot of the really nice the section work that you saw with the lamination, also all the synaptic work. Ruchira Singh did all the Best disease work along with Wei Shen, who now works for Pfizer. A lot of the really early stuff that I showed you was done when Jason Meyer was here, who's now an assistant professor at IUPUI. And then all these folks in here contributed in many different ways. So I thank you very much for your attention, and happy to answer any questions.
Thank you very much for a very in-depth, enlightening discussion. Let me ask you something just about Best disease. I mean, as you stated, the bestrophin is in the respiratory epithelium. These patients don't have respiratory.
You can in culture see defects, but they don't. You're right. They don't come in and gasping like CF or something like that. It all goes back to what we were talking about before with gyrate atrophy. There's plenty of genes that either have redundancy in some tissue and not in another, such that if there's nothing to pick up the slack.
Or is the epigenetic profile different in respiratory epithelium from the RPE?
That's a possibility too. It kills me that we all have to talk about epigenetics now because it's complicated enough without having to worry about that. But we're actually that is something that we're looking at more from a development standpoint. But yeah, I mean that easily could be invoked as well.
The other thing you mentioned at the very beginning is that the RPE isn't necessarily affected by the bestrophin defect at the outset.
Yeah.
But what is it ultimately? The accumulation of A2E that ultimately results in the RPE
being dysfunctional.
Being dysfunctional and then dying?
No. If you look at RPE and histopathology in 80-year-olds with bestrophin, the RPE is still there. So you don't see degenerated RPE for the most part. This is the histopathology studies that are available out there. So you don't see like a geographic atrophy where the RPE is gone or dead.
Don't you see metaplasia of the RPE? I mean, isn't there fibrosis?
Bony sort of thing. I mean, you get gliosis. You can get gliosis, right? But you'll get that without a primary RPE defect. You get gliosis in primary photoreceptor degenerations. And you do have a secondary to photoreceptor degeneration. So that's going to be a Mueller-glial or a microglial reaction. So the neuroretinal effect would do that.
These IP-derived RP cells are different clones, aren't they? Not a single clone.
So they're all a single clone.
So you start from a single cell?
A single, in terms of clone colony.
No, I'm talking about you expand a single clone. Because
so it's impossible to know that for sure, because when you make the clones, you spread them out, you dissociate them all out, you put them on a plate, and then when you first see them, they're a clump of like 3, 4, 5 little cells.
Pick one cell and then
And then you pick that little clump.
can you dissociate and then get a clone?
If you dissociate ES and iPS cells, they won't grow. They need buddies next to them. It's very hard to clonally derive an ES or iPS cell. I don't think anybody's really ever done it.
Because you know, I mean, normally in natural adult RP cells have different cell populations. They're actually
In different what?
different cell populations.
Okay.
One RP next to
Yeah.
the other RP is the same. They have different metabolic activities, not only topography. Their catalase activity is different. Their lysosomal enzyme
Right.
activity is different. I wonder whether, if it's not a clonal thing, how much it represents the individual RP cells in the body.
Well, but you could say the same thing about an animal model, right?
Sure.
I mean,
Animal
right? I mean,
model
anything.
represents-
So I think the point being is that nothing is perfect, right? You can't, and that's why I went to such lengths at the beginning to talk about the fact that you can pick apart every single model. A mouse doesn't have a macula, right? So you have to know those limitations, and I think there are some bigger ones and there are some lesser ones, and you have to meld that in with the other available. I mean, this is by no means. I think every paper I've ever written has always said this is not meant, because people get so sensitive about it. It's not meant to supplant animal model systems. It's not meant to supplant observation of patients, which are all critical. But yes, there are things that you say, you know what, is that really real? Is that not really real? And it's levels of probability, ultimately.
If you tell me which is going to have the most important medical application, to take one iPS cell and transfect it Avastin gene and put it back in the subretinal space.
So to deliver avastin? Actually, Peijuan Guo is, well, this nanoparticles might very well be the way to go with that too. So pRNA, I think he's working on that, and that looks pretty good.
Any other questions? Yes.
The best disease model you
Mhm.
have with the calcium emission, is it the capacitated calcium entry affected, or because the STIM1 is involved in capacitated calcium entry
Across the plasma membrane.
Yeah.
Right. Well, there is the ATP response involves release at the endoplasmic reticulum, but also involves changes at the basolateral membrane as well. So, but again, you're right. Is that specifically important for the? I mean, that membrane fractionation experiment was not had anything to do with calcium. That was just a membrane fractionation experiment. So that may have nothing to do with the ATP response. If I'm answering your question correctly.
Yes, but can you not measure the store size within the cell without ATP?
Yes, so the absolute, so basal levels with Fura2,
And also just opening the capacitated calcium channel and have more calcium coming.
as part of a perforated patch clamp. Is that what you're talking about? Because yeah, we don't have that data right now. But yes, that's something you can do. And one of the nice things about working with the RPE is that you can do some relatively, in-depth electrophysiology on them as well. So our problem is, it seems, is knowing kind of where to stop a little bit too, because you can follow things forever, and we're trying to say, okay, well, we haven't touched on. I didn't show you any of the pH data either. It's much more subtle than the calcium data. We're not seeing subtle differences, but not. But in my mind, that's not a bad thing, because I come at it with a skeptical eye too, because you are developing an artificial system in a culture. No more so than, and you have to realize too that most of the other work that's done, how is it done? Overexpressing cells in COS and HEX cells. So most of what we know about Best disease is done through overexpression systems in non-RPE cells. So again, what is your comparison? What I mean, is this a step up from what's available or not? And I got myself off tangent. Now I don't know what you were talking about, which is you should all be thankful for that.
And that's a good point for us to stop. Thank you very much.
Thank you very much. I appreciate being invited.